Okay, good morning, everybody, and welcome to virtual seminars for Precambrian geology. So today we have a talk by Tina Woltz from UC Santa Barbara, assessing preservation bias in the Proterozoic record of organic walled microfossils in shale. So we're very excited to have our first PhD student giving a talk today. And we hope to have more in the future, more PhD students and postdocs. We've set up our schedule all the way through 2021, but I'm going to set things up for people to start proposing more talks and nominating other speakers. And hopefully we can do that through another Google form on the website. It's not there yet, but it will be. So if you have some good ideas for people who should speak, then in a few weeks, hopefully I'll have that set up. Next week, we have a talk by Alessandro Ielpi from Laurentian University titled Bridging Geomorphology and Precambrian Geology, a broad view of the Precambrian fluvial record and why modern hinterland basins matter. So more in the realm of sedimentology and trying to connect the modern and the past. So join for that as well. And with that, I'll stop talking, and Dr. Susanna Porter from UC Santa Barbara, Tina's advisor, will go ahead and introduce Tina for us.
Thanks, Alex. So Tina came to UCSB from University of Utah, where she got her Bachelor's of Science degree in 2016 from the College of Mines and Earth Sciences, and she was their top student, won the department Best Student Prize. Since coming to work with me, she has not only been great as a research student, but also ended up being basically our top teaching assistant, and was elected by the faculty to be our sort of lead teaching liaison. So she's talented in many ways. This project is two parts of her PhD thesis and a third project on the Uinta Mountain Group Microfossils from Utah. She's finishing up this year, so this is her last year, this next year, and then on to greater things. So Tina, go ahead and take it away.
Great. Thanks, Susanna, and thanks, Alex and Andre, for organizing this seminar. So today I will be talking about preservation bias in the Proterozoic record of organic walled microfossils in shale. And I'd like to start off by thanking my many collaborators, especially Ross Anderson at the University of Oxford and Nicholas Tuska, now at Cambridge, who've been instrumental in some of the work I'll talk about today on clay mineralogy. So let's start by talking about what an organic walled microfossil is. The name kind of gives it away. It's any microscopic fossil that is composed of organic matter. And there are a variety of forms. There are spherical forms shown here, filamentous, colonial, and some forms have detailed surface structures. There are processes that come away from the vesicle surface in this example, and here there is grooves and ridges that are distributed across the surface, the fossil surface. And these more complex structures are interpreted to be the cells, the active cells, or the resting cysts of eukaryotes. And so these organic walled microfossils record major events in the early history of eukaryotes. This is a recent review by Phoebe Cohen and Francis McDonald that show the diversity within assemblages through time through the Mesoproterozoic to the end of the Neoproterozoic. And the width of these horizontal lines represent the age uncertainty in these units. And so these microfossils record the first appearance in the fossil record of eukaryotes, a broad, low diversity across the Mesoproterozoic, and a rise in diversity in the late Tonian. this broad pattern is corroborated by another study that focuses in on the late Tonian. Again, this is the number of species present through time, but time is shortened, showing only from 800 million years ago to 720. And again, we see a rise in microfossil diversity, followed by a decrease in diversity about 10 million years in advance of the global cryogenian glaciations. So it's encouraging that these two recent studies come to the same conclusion, show the same general patterns in microfossil diversity through time, but it's also important to consider what biases this record might contain. As you might have noticed, there is a sampling bias. There are much fewer horizontal bars in the Mesoproterozoic compared to the Neoproterozoic. This can be seen more clearly in these box and whisker plots, showing, again, the diversity on the Y and time broken down into categories. There are half as many fossiliferous units described in the Mesoproterozoic compared to the Tonian. And you'd have to remember that the Mesoproterozoic is twice as long. And so there's way fewer samples or assemblages described. And this is concerning when looking at the median diversity of these units. This is this central horizontal bar. They're quite similar from the Mesoproterozoic to the Tonian, and this rise in Tonian diversity actually comes from these outliers. So given that the Tonian is so much better sampled, it's possible that there is actually a higher diversity in the Mesoproterozoic, but just purely by chance, it has not been sampled. Apart from sampling bias, there are also preservational biases. These can be caused by, these are large-scale variations in preservational regimes that affect fossil diversity patterns through time or ecological patterns. And they can be caused by a variety of phenomena, including climatic or tectonic changes or changes in biology, either the appearance of a new taxonomic group or changes in the behavior of an existing group. These have been very well studied in the Phanerozoic, and some, of course, have been studied in the Precambrian. But it's important to understand these preservational windows in the Precambrian because each window, although it gives a unique view of life at a certain time, there are always biases associated with them. So let's first consider where we find Precambrian organic walled microfossils. They're found in shale, chert, carbonate, and phosphorite. If we scale these boxes to be proportional to the number of fossil-bearing units, however, it's very clear that the majority come from shale. And for that reason, it's important to understand how these organic walled microfossils are preserved in shale to assess if there are preservation biases within this record. So I've spent the majority of my time as a graduate student considering how organic walled microfossils are preserved in shale, but this wasn't the original plan. So I started my graduate work thinking I would study and describe the fossil assemblage of the Uinta Mountain group. And in the summer before I started my PhD, I met up with Susanna and Carol Daler at Utah State University to do some field work in the Uinta Mountain groups. And it's in Utah, so it's close by. I was living in Salt Lake at the time, and I was very excited. First field work of graduate school. But I really didn't know what I was doing when it came to sampling shales, especially choosing shales that are likely to yield microfossils. And it was only through Susanna that I learned that there is this general rule in Precambrian paleontology that to sample light green and gray shale, because this roughly corresponds to low concentrations of organic carbon. High shales with high amounts of organic matter usually don't contain microfossils, and when they do, they're very poorly preserved. So this is general knowledge in Precambrian paleontology, but it's only from anecdotes small mentions in papers and advice from advisors, and it hasn't been rigorously shown. So in any case, I moved to California and we send these samples from our fieldwork off to be macerated in hydrofluoric acid. And this results in the organic residue that we can then observe under light microscopy and identify fossils. And so the results from these macerations were pretty mixed. There were some beautifully preserved microfossils present, but there was also a variety of preservational quality. There were some samples that were dominated by very poorly preserved microfossils. And so from here, I paused my work on the Uinta Mountain group, and I got interested in questions on preservation of organic world microfossils. And in particular, I wanted to characterize the relationship between total organic matter measured by total organic carbon and fossil preservation. Like I mentioned, this was already known in my field, but only anecdotally. So I wanted to quantitatively measure this relationship. So I tested if fossil presence or absence and preservational quality varied with amounts of total organic carbon. Given that we know that total organic carbon varies significantly from sample to sample, I also wanted to know if total organic carbon might affect fossil diversity patterns through space and through time. I also hope that this work can be used by paleontologists to target shale samples that are most likely to contain microfossils. And so our data include 346 samples from 4 paleo continents and have ages that range from late Paleoproterozoic to late Neoproterozoic, with the majority of ages falling within the Tonian. First, let's break down our results by fossil presence and absence. 189 of the samples were fossiliferous, 157 were barren. And this is a probability density function showing the distribution of total organic carbon broken down between those with fossils and those without fossils. And this is a way to visually show that in samples with low amounts of total organic carbon, you're relatively more likely to encounter a shale with microfossils compared to those with higher amounts of total organic carbon. So to test if this is a significant relationship, if this distribution that we're seeing might be produced by chance, we randomly subsampled our data. We took 189 random subsamples from our data representing the number of actual fossiliferous samples we had and recorded the median. We repeated this process 10 to the 5th times to produce this histogram of median TOC values from random subsamples. And given that the actual median of our data of fossiliferous samples falls outside of the 95% confidence interval of all these random subsamples, it's unlikely that we have such a low median of fossiliferous, of TOC values in fossiliferous samples purely by chance. Next, we compare preservational quality to total organic carbon. This is done by ranking the preservational quality on 3 indices of preservational quality: the loss of surface pattern, the density of pitting, and the degradation of wall margin. Each microfossil was ranked on a scale of 3 corresponding to excellent preservation and 1 corresponding to very poor preservation. Within each sample, 50 randomly selected fossils were ranked on each index of preservational quality. I should note, though, that surface pattern only applies to certain taxa with surface patterns present. So if you turn your attention to this image, if you squint, you might be able to see that it's covered in all these organic spines coming from its surface. This is an example of a taxon with surface pattern that can be ranked on this index. These others are Leosphrida, they're smooth-walled, and so they were not ranked on this index. So you'll see in the coming graphs that there are fewer data points for this index. These top graphs show the fraction of well-preserved microfossils, so the fraction of rank 3 within a sample compared to the total organic carbon of that sample. This is compared in all 3 indices of preservational quality. As I mentioned before, this loss of surface pattern naturally have fewer data. And we see a general decreasing relationship between the fraction of well-preserved microfossils with increasing total organic carbon. I think the key here is that there is a lot of scatter, of course, especially in low total organic carbon. But I think the key here is that there is an absence of well-preserved microfossils in samples with high amounts of total organic carbon. These lower graphs plot the fraction of poorly preserved microfossils within a sample, so the fraction of rank 3 compared to total organic carbon. And there's a lot more scatter in these, but again, a general increasing relationship between the number of poorly preserved microfossils within a sample and total organic carbon within that sample. So how do these results affect fossil trends through a stratigraphic section? This is the Chuar group from the Grand Canyon of Arizona. It is correlative with the Uinta Mountain group that I talked about earlier. And so these graphs plot paleontological data through the section. These purple diamonds represent diverse organic walled microfossil assemblages. And these diverse assemblages are seen in samples of different water depths from paratidal to distal subtidal. They're present all throughout the lower formation, the Galeras formation, and into the Awadabi member of the Quagunt formation, and then disappear about midway through this member. This is an example of some of the diverse microfossils that might compose these assemblages. And so at this point, in the middle of the Awadabi member, the disappearance of these diverse organic walled microfossils coincides with the appearance of vase-shaped microfossils. These are teardrop, three-dimensionally preserved shells or tests thought to be from amoebae, and they're preserved in a variety of different preservational pathways. So it's likely that these are preserved differently. But in any case, this shift in the assemblage is considered to be abiotic. It's interpreted to be a biotic turnover event. However, right across this interval, total organic carbon concentrations are also increasing. So it's at least possible that preservational quality might affect the appearance of this pattern. We can plot the fraction of well-preserved microfossils in accordance with the pitting density index and the margin quality index through the stratigraphic column. And you might notice there is a lot of scatter in the lower formation. That's to be expected, but there are still some well-preserved assemblages. And the part that I find most interesting is at the top here, where there is a decrease in the proportions of well-preserved microfossils present. And this coincides with increasing total organic carbon and this biotic turnover event. And this is not to say that there was not any biologic change at this time, but changes in preservational quality can affect the appearance and timing of these biotic changes. Next, we can consider how changes in TOC, total organic carbon, can affect large-scale patterns. This is a compilation of total organic carbon through the Paleo Proterozoic to now, the present. And the majority of these data come from Sperling and a recent paper by Sperling and Stocky. The median TOC values within each grouping is reported at the top, along with the number of observations of TOC measurements within each grouping. And my eye goes straight to the Neoproterozoic. This interval has anomalously low total organic carbon when compared to the Mesoproterozoic and to the Paleozoic, excuse me. And at this time, this is the time interval where we see a rise in eukaryotic diversity. And so provided that eukaryotic diversity increases at this time, this work at least raises the question of whether this diversification could be attributed to better preservational conditions compared to that of the Mesoproterozoic, for example. Also, going forward, I hope that this work can be useful to paleontologists who are looking to study new units. Of course, as I mentioned before, this pattern between total organic carbon and fossil preservation was already well known and utilized in my field, but I hope that I've helped to constrain this idea a little bit more. And I hope it can be useful for those paleontologists studying shale from drill core, as these drill cores are usually drilled by oil companies that regularly measure total organic carbon. And so there is an abundance of data at our fingertips that we can use to our advantage. And so we can use that TOC value, those TOC values, to target shales that at least have a higher potential of containing well-preserved microfossils. So I hope at this point I've convinced you that we do see a pattern between total organic carbon and fossil preservational quality. I haven't talked quite yet about what could be causing this relationship. So now let's consider one idea, and that is that clay minerals can play a role in organic preservation, organic fossil preservation. There is a huge body of work on the interactions between clay minerals and organic compounds. And just to highlight a few, this study that takes modern sediment from the coast of Washington plots the surface area of the sediment against the amount of organic carbon that is bound to its surfaces. And if you focus in on these points labeled as C's, this is the clay fraction of the sediment. And you see that it has a high surface area and that it preserves quite a lot of organic carbon on its surfaces. This is another study from topsoil that visually shows that carbon and organic carbon and clay minerals clump together in this elemental map. The blue points are the clays and the yellow is organic matter. And you see that they are in a direct association with each other. This body of research about clay organic interactions has not gone unnoticed by paleontologists. Many have applied this knowledge to help explain how organic fossils are preserved in shale. About 20 years ago, work by Nicholas Butterfield suggested that clay minerals are a principal control on organic fossil preservation. This cartoon depicts an organic vesicle, so the remains of some microorganism that, fingers crossed, if all the conditions are right, might become a fossil. This is a schematic of a depositional environment with clay minerals available and around. Different types of organic matter, amorphous organic matter. These can be broken down components of other cells or the waste products of other cells. And since we're never in a sterile environment, there are degradative enzymes around too. These can be produced by degradative bacteria, or they can be produced by the vesicle itself upon death. These enzymes that are used for housekeeping are released into the environment. So these are the main players. And in this end member scenario, the concentration of organic carbon to clay mineral content is low, so that there is plenty of reactive clay mineral surfaces to bind to amorphous organic matter and these degradative enzymes. And in binding to these degradative enzymes, they become sometimes temporarily deactivated or other times permanently deactivated. And so they're no longer capable of degrading the organic remains, this organic vesicle here, for example. You can imagine the other side of this, the other end member, when there are high concentrations of organic matter to clay mineral content, there's less clay mineral surfaces available for these reactions between degradative enzymes and clay minerals, so that some are uninhibited and are able to degrade the organic remains around them. In this case, we would expect preservation to be poor, or maybe in some cases, the organic vesicle would not be preserved at all. This was first proposed in the context of Burgess Shale type preservation and has since been suggested for certain exceptionally preserved organic walled microfossil assemblages like the Svanberg Phyllite Formation. Several laboratory experiments support this idea. In this experiment, the growth of this degradative bacteria was measured in the presence of different concentrations of calcite and clays, and compared to a control where no substrate was added. And with increasing the amount of clays, this decreases the growth. This significantly limits the growth of these degradative bacteria. The study also finds that certain clays are especially effective at bringing down the growth of these bacteria. Again, this is the same y-axis, and we can see that berthierine and kaolinite limit the growth of these degradative bacteria more so than in samples with calcite, glauconite, and illite added, for example. So because clay minerals are so reactive, there are other ideas, of course, on how clay minerals might affect fossil preservation. Because of the platy shape, the high surface area, and the high surface charge, it's possible that clay minerals bind directly to the organic vesicle surface, thereby protecting it from degradation. And in some cases, it's been suggested that this association actually changes the chemistry of the vesicle surface to become more resistant, into more resistant and stable forms, increasing the preservation in that way. There's evidence for this as well from laboratory experiments. This is an incubation of a lobster egg in the presence of fine-grained sediment. And you see after 10 months, there is a coating of that material all across the surface of this lobster egg. This is a close-up image of that surface showing all the particles, and a cross-section showing that indeed there is a thick crust on this egg. Other experiments have buried annelid worms in in sediment of different chemistries like kaolinite, calcite, quartz, and montmorillonite. And after 4 months, this material is then tumbled around to see how intact the organic remains are. The results are shown in these petri dishes below. And kaolinite, after 4 months, still has a relatively well-preserved annelid worm present. It's pictured here. The authors suggest that kaolinite aided in the chemical alteration of these structures to aid in their preservation. There is further evidence that comes from direct observations of Burgess Shale type assemblages. This is a principal coordinate analysis that visually shows that the mineralogy of samples that contain Burgess Shale type fossils is significantly different than the mineralogy that contains mineralized skeletons, something, non-exceptional preservation, so that there's a specific clay mineral assemblage associated with Burgess Shale type deposits. The probability of a sample to contain a Burgess Shale type fossil was also found to be positively correlated with berthierine concentrations and negatively correlated with sedimentary and illite concentrations. Further work shows, measures this in situ. These are SEM images of Atoya and Opabinia, and these highlighted gray boxes are where in situ X-ray diffraction was performed to get the mineral assemblage present. These elemental maps of the same fossils show that the elements present on the fossil surfaces are different from the matrix that surrounds it, and results from the X-ray diffraction measurements show that the surfaces of the microfossils are elevated in kaolinite. And so kaolinite is thought to play an important role early in the preservation of these soft-bodied organisms. There is also limited evidence in the Proterozoic for microfossils being preserved by specific clay minerals. This is Siphonophycus from the Wynyatt. The microfossil is this dark line, and it's surrounded, you might have to squint to see this, but it's surrounded by this platy halo that's only about half a micron in thickness. This is another example of Siphonophycus that is cross-sectioned and the elemental abundances are mapped. This carbon film here is the fossil itself, and it's surrounded by, it's enriched by aluminum right in its vicinity. These minerals here are interpreted to be kaolinite. A similar study cross-sectioned this microfossil and mapped its elemental abundances, finding that there are elevated levels of iron on the outside of this microfossil and potassium inside the vesicle. These correspond to berthierine concentrations and illite concentrations, respectively. So given that there is so much evidence for the role of clay minerals in fossil preservation, both in Burgess Shale type preservation and some evidence in the Proterozoic, I wanted to test if clay mineralogy plays a broader role in organic walled microfossil preservation, and if it can help explain the huge variability in preservational quality that we've been seeing, and that I showed you in the previous work, in the previous slides. So we measured the bulk clay mineralogy for 232 shale samples that range in age from Mesoproterozoic to late Neoproterozoic, again with the majority being Tonian in age. And about half of these samples were barren. And for the interest of this talk, I will only be focusing in on the 78 samples for which fossil preservational quality was assessed. So first, we identified the minerals present within each shale. This was done by powder X-ray diffraction, which scanned over 5 to 80 degrees of 2 theta. And we also took an additional scan of this region here between 69 and 75 degrees of 2 theta, because in this region, there are broad peaks of clay minerals that are distinct from one another. And these peak signals, yes, are characteristic of specific clay minerals. And the area under these curves are proportional to their concentrations within the sample. Again, we compared this to preservational quality. We focused in on just 2 of the indices, the density of pitting and the degradation of wall margin. Again, we ranked each fossil on a scale of 3 to 1, corresponding to excellent preservation and poor preservation. And again, 50 randomly selected fossils were ranked on each index. So these are data you're already familiar with. This is the fraction of well-preserved microfossils within a sample compared to the amount of total organic carbon within the sample, according to these two indices of preservational quality, the density of pitting, the degradation of wall margin. And I should note that each point is a sample. And so each point has, is made up of this fraction of, that is made from 50 fossil observations. So in total, given that there are 75 data points, that adds up to be about over 2,000 individual fossil observations. So in any case, we do see this negative relationship between well-preserved microfossils and total organic carbon, comparing just the total amount of clay that we see within these shales, comparing this to the fraction of well-preserved microfossils, there isn't a very clear pattern. There might be a very weak positive relationship, but there is a lot of variability here. Now, if you remember back, one of the predictions of the hypothesis that clay minerals inhibit these free degradative enzymes is that the ratio of organic carbon to the total clay is important, and that low concentrations, low ratios would yield well-preserved fossil assemblages. So here we did just that. We took the ratio of total organic carbon to the total clay content within a sample. And there is a negative relationship between the fraction of well-preserved microfossils and this ratio. It explains more of the variation in preservational quality as well. Using these raw data, we can make multiple logistic regression, basically taking these data and using this ratio of total organic carbon to clay to predict the probability of finding a well-preserved microfossil, both in accordance to these two indices of preservational quality. And several tests, the log likelihood test and an Akake information criterion, indicates that the ratio of total organic carbon to clay is a better fit to these data than total organic carbon alone. And so these tests justify the inclusion of this extra variable to help refine this relationship. We can also look at specific units, like the Chuar group from Arizona, for example. This is plotting, again, the fraction of well-preserved microfossils to this ratio of total organic carbon to clay. And this relationship, this negative relationship is present even within this unit. And then when considering that some members of this unit have experienced higher thermal maturity, their past peak maturity, these are highlighted in, or these are muted points, this relationship becomes even stronger. Next, as we've seen from previous studies, clay minerals, different clay minerals react in different ways. And so we compare the probability of finding a well-preserved microfossil to the concentrations of kaolinite, illite, and berthierine. These black marks along the x-axis represent where our samples are in relationship to their concentration of these minerals. And we find that kaolinite and berthierine have negative relationships with the probability of finding a well-preserved microfossil, and illite has a positive relationship. Next, we can look at this other index of preservational quality, and this shows similar trends, a negative relationship again with kaolinite and berthierine, and a positive one with illite. So although these clays were found to significantly affect the probability of finding a well-preserved microfossil, they explain very little of the total variation in preservational quality that we see within these samples. If we take illite, for example, illite concentrations range anywhere from 0% of the sample to about 80% of the sample. But this increase from 0 to 80% only increases the probability of finding a well-preserved microfossil by about 20%. And that's really not a lot. That's not really a huge change, especially when compared to how much variation is seen within these samples and how much variation is explained more simply just by the ratio of total organic carbon to total clay. So to sum up our results, we found a significant relationship between fossil preservational quality and the ratio of total organic carbon to clay content. And we found significant but rather weak relationships with preservational quality and specific clay mineral concentrations. What can these data tell us about the preservational pathways of these organic walled microfossils? And in particular, are they similar to what is seen in Burgess Shale type deposits? There are a few reasons to think that organic walled microfossils are not preserved in the same way as Burgess Shale type fossils. For one thing, the Burgess Shale preserves very soft tissue of animals, and these are very delicate and were likely highly reactive. In this case, a fossilized gut is preserved, and this is illustrated here just to show that Burgess Shale, these early animals had guts, and within these guts there was an active microbial community. Degradative bacteria were present, and so upon burial, this community is surrounded by labile organic matter that it can then consume. And so in my mind, it makes sense that Burgess Shale type fossils would need very specific conditions in order to be preserved, very specific clay mineral assemblages, for example. And that is exactly what some data show. Again, this is a study by Ross Anderson et al., showing that there is a distinct and specific clay mineral assemblages associated with Burgess Shale type fossils. And I want to draw your attention to this plot over here that shows positive relationships with berthierine and negative relationships with illite. And as a reminder, we found the opposite relationships. We found a negative relationship between berthierine and the probability of finding a well-preserved organic walled microfossil, and a positive relationship with illite. This further suggests that organic walled microfossils are preserved by different conditions to the Burgess Shale type fossils. So in comparison with Burgess Shale, organic walled microfossils are considered to be much more resistant. They are often compared to the cysts of dinoflagellates or the cell walls of chlorophyte algae. And these structures, are built to withstand adverse environmental conditions, including degradation. And so it's possible that these organic walled microfossils do not need specialized conditions in order to become preserved. On the other hand, there is some evidence from previous studies that there are interactions, or at least associations, between specific minerals and some units that preserve organic walled microfossils. This is Proterozoic, for example, from the Svanberg fielet, which is very thin and delicately preserved. Its thickness is on the order of tens of nanometers in comparison to these other vesicles that are hundreds of nanometers to 1 or 2 microns in thickness. And there are others. This is Siphonophycus from the Winiot that again show an association with a specific mineralogy. So there's a few possibilities to explain this difference. One is that there are several different ways in which organic walled microfossils might be preserved in shale. Some may need specialized preservational conditions. Another possibility is that the bulk analyses done in this study do not measure the importance of these specific clay minerals, especially at these small spatial scales in which they're interacting. you can imagine here in this SEM, this halo is only 0.5 micron in thickness. And that's not a lot of material needed in order to completely coat this microfossil. And so maybe it's even though it's important at these small spatial scales, the abundance of that mineral within a sample might not show that importance. So what role do clay minerals play in preservation? My data are not inconsistent with any of these hypotheses, and so I can't reject any of them. That might not be a very satisfying result for you all. But in any case, they're also not mutually exclusive. It's possible that several of these are interactions are happening in conjunction. But in thinking more about the results of this study, of my study, that show that there is that the ratio of total organic carbon to total clay content is a good predictor of preservational quality. One possibility is that preservation is simply governed by simple diffusion. In this diagram, there is a bacterium at the center producing extracellular enzymes. So these enzymes that degrade organic matter. And these squiggly lines are how it's diffusing away from where it's produced. It's living in sediment, and also within the sediment is organic matter that's being broken up over time, and the broken up products are diffusing into the environment. And in environments where the ratio of organic carbon to total clay content is low, organic matter becomes physically separated from the enzymes that degrade it or the bacteria that degrade it. Increases in clay content, since they're platy and usually small in size, would reduce the porosity, which would further limit the diffusion of these enzymes. And so together, low amounts of organic carbon and high amounts of clay would reduce the diffusion and would both separate out these organic pockets from one another, similar to what a food desert. And higher amounts of clay would reduce the diffusion of these enzymes to these organic pockets within the sediment. Great, so this is just summarizing what I've said into words. High concentrations of clay minerals would limit the diffusion of enzymes, and lower concentrations of organic matter become physically separated from the degradative enzymes. So with that, I'll leave you with some final thoughts. Total organic carbon is negatively correlated with preservational quality, and the ratio of total organic carbon to clay content is an even stronger predictor of preservational quality. The bulk mineral analyses show that preservational quality have weak relationships with certain clay minerals like berthierine and illite, and that given that there aren't very strong relationships between these specific clay minerals and fossil preservation, we can have a little bit more confidence in the Proterozoic microfossil record when it comes to differences in sediment compositions. However, it seems that total organic carbon can still be used as a tool to identify to identify sediments that are likely to contain organic world microfossils. So with that, I'll turn it back over to Alex for questions.
Great, thank you so much, Tina. Fascinating and well delivered. Great, so we are going to open up for our question session, which you can approach in a couple of ways. There is a reaction for raising your hand in Zoom, virtually raising your hand, and you can, if you can't find that, you can also just type a star into the chat, and I'll address questions as they're coming in order that they're received. Alternatively, if you don't have a microphone, you could also just type your question in the chat and I can ask it for you. So Eric Sperling says, great talk, Tina. So cool to see this deep dive into the role of TOC and clays and fossil preservation.
Thanks, Eric.
Hi, Christina. Maybe I just start with a quick question. So you talk about illite. Illite typically form later, but with fluids transporting potassium. Do you separate? Do you mean detrital illite, or do you mean later formed illite? And if it's a later formed illite, could be a role of these fluids in degrading organic matter, microfossils.
Yeah, Andre, that's a good point. These bulk analyses don't discriminate between illite that were present to begin with and those that are a result of later diagenesis. That is something to consider, and maybe a deep dive into specific formations or specific units can help with that problem. Some that have maybe better constrained thermal histories.
But probably if it's detrital illite, it would not be reactive anyway, so it will not help to preserve microfossils. And if it's later illite, then it might help to destroy microfossils. So both type of illites probably will act against preservation.
Will act against preservation. Huh. Yeah, that's something to consider. I haven't thought about how the different types of illite and how that might affect fossil preservation. However, we do see a very weak positive relationship with illite, and I'm not exactly sure why that could be. It might just be, yeah, the slopes, as you saw for those lines, were pretty low, and so maybe illite isn't playing a significant role.
Maybe it's a role of precursor to illite.
Yeah.
Can I jump in here to a great talk, Tina? I really enjoyed it. There is a way to tell late diagenetic from early illite, and that is by the narrowness of the 10 angstrom peak. Something that's been used in rocks for years to look at metamorphic grade. So you probably have it if you went all the way from 0 to 80. If you look down at the 10 angstrom peak and you measure the peak width, which is the Kubler index, or the half height to the total height, which is the Weaver index.
Yes, that's a good point. And I should have said I have looked a little bit more closely in these X-ray diffraction patterns and looking in the different types of illites, the 2M1 illites, the detrital signal illites compared to total illites. And as far as I can see, there is no relationship between those specific detrital illites to preservational quality, which is an interesting detail.
All right, so we have a question from Don Davis. Great talk. How might clay minerals migrate to the fossil?
Oh, well, this might, let's see. So there are a few thoughts on what kinds of minerals interact with the fossils. Some are shown like that study I showed of the lobster egg. Those were detrital minerals on the surface of these lobster eggs. But there's also evidence that suggests that these clay minerals might nucleate directly on the surfaces, so form on the organic matter itself. And I think just with compaction and natural movement of sediment, early on, these clay minerals can form this crust on the fossils.
Great. We have another question from Ava Stoikin. Hi, Christina, you may have mentioned this, but do you see differences in preservation between different paleo environments or sedimentary facies?
Oh, that's a great question. And that's actually something I'm looking into a little bit more. It can be tricky when total organic carbon varies from, shallow peritidal environments to more distal environments. If there's a trend in total organic carbon, it's difficult to pull these effects apart. But I found within paleo environments that there is still a pattern between preservational quality and total organic carbon, and there is a trend between at least total organic carbon and these relative water depths, as I showed from the Chuar group, with higher preservation in the shallow environments. That's a very good question. Something to definitely consider more.
Great. We've got another question from someone named SX. Sorry, I don't know. Oh, Shuhai Zhao. Hey there.
Yeah, thanks. Great talk, Tina.
Hi.
Yeah, I just have a question about this. I think the conclusion is very solid, but I just have this back mind thought. When we extract aquitox from shales, if there's a lot of organic carbon, typically we have to use nitric acid to oxidize some of the organic carbon, so make it easy to see the fossils. So I wonder whether the pattern that you see is also at least partially the result of extraction bias. Namely, if there's too much organic carbon, it's difficult to extract the microfossils or aquitox from the shales. Not because they're not there, but because it's just difficult to take them out because there's too much organic carbon, right? So I wonder whether you've thought about this and how to test this. Obviously, because this is related to the availability of data, it's difficult to test using available data because you don't extract fossil from jet black shales. It's difficult. They are there, coal beds, for example, there are a lot of pollens and things like that. If you take the extra step to extract them. Maybe play the role. I'm not saying that, I think you have demonstrated very conclusively there's a relationship between TOC and the clay and the preservation. But on top of what you have said, whether there's other factors that are also important and they need to be considered.
Mm-hmm. Yeah, it's a very good point. And I have considered the possibility that in these organic rich shale, there's just so much material to look through that maybe you're just not encountering as many microfossils just because of the abundance of organic matter present that kind of hides these microfossils. And I've thought about ways to test this. One would be spiking the samples in maceration with, lycopod spores or something that you can identify, and then counting the number of fossils found relative to the abundance of organic matter present, kind of controlling the amount of material that we're looking at. And we have preliminary data from fossil to organic matter counts showing that there are, if you randomly point count 200 points within a slide, let's say, there are relatively fewer organic world microfossils to compare to the amorphous, junky organic matter that's also present. But it is something to consider, and hopefully I can do more work on that.
Yeah. The quick question is, you made an interesting comment, the correlation of eukaryotic diversity and TOC that is compiled, the big picture, Paleo, Proterozoic, Neo, and Paleozoic. And you see the low TOC in the Neoproterozoic, and you made a comment that there's a rise in diversity in the Tonian, but that diversity increase is largely driven almost singularly by the 15-mile group that is not organic world microfossils. And in contrast, when you look at the Paleozoic, TOC, the compilation, TOC is very high in the Paleozoic. But when you look at Ordovician, the aquitach is just, and if you look at, some of the Ordovician shales, the number of aquitach is just dwarf anything you see in the Neoproterozoic. So I think you need to, that general pattern may not tell us a lot about the correlation between TOC and diversity. One has to, like you did, has to look at the individual successions.
Yes, that's a good point. And I mainly wanted to use those graphs to compare the Mesoproterozoic to the Neoproterozoic. Like you said, there are organic rich shale and the Ordovician, Devonian that have an amazing diversity of microfossils and also are high in organic carbon. So since none of my samples are from the Phanerozoic, I can't really speak to how they're preserved. Potentially they're preserved in a different way, which would be an exciting other new project.
Okay, great talk. Thanks, Tina.
Thank you, Shuhai.
All right, we have another question from Franco. So it's considering that sedimentary, berthierine, and kaolinite are very sensitive to the geothermal gradient, do you think the actual abundance on these clays are still representative of the primary composition in order to attempt a solid correlation with fossil preservation?
To be honest, yeah, I don't know when these minerals formed. They might be a product of later thermal alteration, or they could be a product of nucleation and formation early in early diagenesis. I'm not certain. It's a difficulty of this clay mineralogy work.
Mm-hmm. All right, France, you can feel free to unmute yourself if you wanted to follow up on that. But other than that, we don't have any more questions on the palette here, or I don't see anybody raising their hand. So I can give people a few more minutes, or a few more seconds to do so.
I think France implied that berthierine and kaolinite can easily be altered with geothermal gradient, and so this way it would decouple preservation of microfossils and clay mineralogy that we see now. Do I understand correctly your question, Franz?
Yes, Andre, yes.
So basically he's saying that it could have been much more berthierine and kaolinite early on, but now they transferred to different minerals.
Oh, I see. I understand. Thank you for helping clarify. Yes, that is possible. That could possibly be taking place. I think some of the in situ work that's recently taken place by Ross Anderson is a good way to get around this problem of berthierine and illite being altered through diagenesis. And maybe that's something I should consider for these samples.
Well, Tina, I think you did a fantastic job today, and thank you very much for speaking today. You guys at Santa Barbara always do such a great job. So anyway, okay, just a comment for France in the chat. But I think we've exhausted. It looks like we've exhausted the question session. So thank you again, everybody, for coming. Thanks for the great talk, and we'll see you next week for another round. All right. Have a good week, everyone.
Thank you.
Bye. Thanks.